to use it. There is a little game to start with where our body is very dynamic. And so if you don't mind, I'm going to try to ask you to start by telling me how the people that you're going to see as silhouette on the screen are feeling. What is their state? So if I show this image, what do you think the people are feeling like? Sorry? Partying, joy, excited, yes. And then if I take this one, partying,
Painful.
sorry?
Painful.
Painful,
Yeah.
sad. What else? What do you think? Maybe sick, I don't know. And then what about this one?
Admiration.
Admiration, curiosity, maybe also desire, don't know. And what about this one? This one is, you see it a lot also in Japan, I must say. Disconnected. And what about this one? Flying. Light. Yeah, and joy also, maybe ecstatic. So indeed, we have like many, intuition for what the body language is, but we don't really have a dictionary for the body language. And so they are like, movements and posture that I just described, but there are also absence of movements. And so I'd like to actually share with you this evidence for, tonic immobility that, maybe people in the audience have witnessed in the ocean, where you can see videos. Oh, actually, you don't see this video. I need to transfer it maybe to the other screen. Can you share my desktop maybe? Or if I don't manage to share it with you, I will have to. Well, don't worry. Don't worry. Okay, I'll just continue. I'll share the videos with the people afterwards. So, so the state of tonic immobility is actually a state that you can see in many different species. In fact, throughout the animal kingdom. So here is an example of an impala pretending to play dead when he's actually under a threat of death from a predator. And by playing dead, he actually managed to escape from being killed right away from the predator. And this actually behavior is sometimes comes out actually very handy. So divers are using this to induce what you see on YouTube as tonic immobility in sharks or hypnosis. It's also called like trance by the divers, where divers are typically putting their hand on the electroreceptor of the nose of the shark. And by doing that, they actually overstimulate the shark and get them to completely lose balance and stop entirely to move. And it's the best way for people to put probes, either by attaching them to the tip of the tail here, or by even like doing a dissection and opening the body to put a sensor in. And so this phenomenon of tonic immobility of sedation is actually really well spread. It's one of the last kind of action that the animal can do when it feels under threat, to completely stop moving when there is no other way out. And the mechanism for it are really not very clearly known. There are also like more chronic change in the body posture that are not dynamic, that like the one we've seen, but that occur with time. And so I don't know if some of you have already witnessed this campocormia, where old people, and it happened, I think, throughout the world for Parkinson patients, but not only, actually losing the ability to stand straight. And instead of standing straight in the ultimate case, they actually stand with an angle of 90 degrees. And what is remarkable about these patients, this is one from the hospital I come from, Petit-Salpêtrière, is that when they stand on their 2 feet, they are forming like, square, like a 90-degree angle in the back. But when you lay them down and they don't have to stand actively, they're actually completely fine and flat. So it's really a dynamic state that comes with the posture being disrupted with age. And there are other more like morphogenetic conditions in which instead of having the typical curvature of the spine, you see people with kyphosis, where you have a high curvature angle here, or lordosis. And if they have 3D torsion, we're going to call this scoliosis. And in this case, it's not a reversible phenomenon. It's really like a static situation that typically happens in the case of scoliosis at the juvenile stage for 8 girls, for 1 boy in teenager time, but also through aging. And the origin for this, both genetic and environmental, are actually very unknown. So if we try to draw a little bit of a schematic of the basic principle for this, if we think now in neuroscience term, we have sensory input that can be both exteroceptive, so they come from outside our body, or they can be interoceptive. And these sensory inputs will largely impact the motor output that can have a postural component and also movement with different actions. This always leads to an efferent copy that will modulate the upstream circuit, often modulating the sensory systems themselves. But in addition to that, the internal states, which are very loosely defined as, all the physical parameters that impact us, the fact that we're hungry, we have thirst, we are aroused, we're anxious, all our emotions, are largely impacting, in fact, this process, so that it can impact the way we perceive things, but sensory perception and sensory inputs also largely impact our state by modulating the activity of neuromodulator in the brain. And so what is interesting is that this, complex mechanism is well, perceived and understood at the psychophysics level, but the cellular basis for it is quite difficult to tackle. And we talked about it on the kind of, transient effect, but there is also a long-term chronic pattern where, the effect of aversive sensory stimulation repeated it over and over again. And we're going to see also possibly pathogen invasion and infection are affecting our internal state. And they can have impact not only in our immediate posture, but also on our morphogenesis. And so it may sound a little strange to use a fish to understand this question, but the really nice appeal from my side, who came from biophysics, like Maya said, is that you have access to every and each of the neurons in the nervous system in the larval zebrafish with about 100,000 neurons that you can image with 2-photon microscopy. The animal is so small, 4 millimeters long, that you can actually look at hundreds of animals doing millions of bouts. And it's very cheap, which, I think is a really important feature when you also think about starting your lab. So the animal is transparent and it's also very fast developing. So you can, take a movie like that with time lapse and look at the development of the embryo, and in 24 hours, the body will form, the essential organs. And you will even see the first motor twitch that you can also observe in utero in mammals. They will occur just after 18 hours in the zebrafish embryo. And so here you start to see that the head will form here, the somite will form there, and the tail will be on this side. And so the eyes are forming here, and then you can see the somite formation there. And when the spinal cord is complete with 30 segments, that's when you start to have the first movements that are really, again, a common feature of all vertebrates, either coming from external fertilization, what Maya studies, or coming from in utero. And so what is nice about this animal is that you can really dive into the brain with 2-photon microscopy and really go look at every cell. But what is also possible, and that I'd like to do even more in the future, is that you can also look you see at the entire animal itself. And so not only focus on the brain cells, but you can also look at how brain cells interact with cells from other organs, because 4 millimeter is totally achievable with fluorescent microscopy now. And so what I'm going to talk to you about is this little sensory cell that is called a CSF contacting neuron that we focus our attention on from some time in the spinal cord. It forms a string of 100 cells in the larva. And those cells are important for interoception. And I will show you how they can connect the sensing of mechanical and chemical cues with the change of posture acutely and chronically. And then we will talk about pathogen invasion, and then we will have a third short story about tonic immobility. So when we started the team at that time, there was, and it's still, I think, the case in the Kindle textbook, that all the mechanosensory information that people were describing as important for proprioception was coming from the periphery with dorsal root ganglia here, bringing information from the muscle spindles, the skin, and the Golgi tendon in order to modulate the activity of motor neurons directly and indirectly in the spinal cord. So during my postdoc, I came across this ciliated cell that looked like disorganized hair cells. And I was quite surprised to see that even though they were conserved and observed for a century, nobody had really investigated their function. And so I went back to the roots of the work of Kolmer and Agder, who describe beautifully with their drawings the presence of these cells in over 100 species. And they had agreed together that it must form a sensory organ in the spinal cord, but they were disagreeing on how they would be sensory. So Kolmer said that these cells must be a third ear in the spinal cord that would sense the mechanical deformation of the spinal cord. And he thought that they would do so by interacting with this black dot here that is a big polymer inside the central canal called the Reisner fiber. And Agder was very much disagreeing with that. He was saying it's an artifact of the fixation. When we get the tissue, at that time, all their samples were coming during the war from farms and zoos. And they were saying it's probably an artifact that they look close to each other, but they probably don't interact with each other. So there were only 2 people at the time interested by these cells, and they were very much already disagreeing. And so and Ernst Reisner described this polymer that they were disagreeing about by just making, at that time in 1860, a single figure done with a pencil and a ruler, where he could draw that in the lamprey spinal cord, in the central canal, there was evidence for an acellular thread that was really straight inside the central canal. And after that, evidence from electronic microscopy showed that you can see it in the rat, in the cow, lamprey, trout, frog, and many vertebrate species. We don't know yet in humans. And in all those species, it's described as this very long protein that is called ethiospondin, almost 5,000 amino acids. And with a red domain here called the hemi domain that's supposed to be important for aggregation, and then a many domain of interaction with LDL receptor and other possible receptors. So we looked at this question with my first student, and we found that the CSF contacting neurons have their little apical extension right by the Reisner fiber in the central canal. And they have, in addition to, what people have described in hair cells of the inner ear, they have also an axon that is ascending in the spinal cord. They are very close to motor neurons in the transection here. And if we look at their activity, we did these experiments with Urs Bohm, we saw that when we tickle the fish by puffing in the inner ear on one side and triggering an escape with a tail bent on the other side, we can monitor their activity depending on whether the fish bends to the left or to the right. And we look at the ratio of the GCAM sensor to the red fluorescent protein to know whether they were activated. And what Urs found doing photon microscopy in 2 wavelengths while recording the behavior is that, in fact, in the wild-type animal, only the cells that are on the side of the contraction, the compressed side, are responding, not the stretched side. And this is absent in the mutant for receptor PKE12-1 that is specific of these neurons. So we had evidence that these cells could be sensing the compression, but the question is, how can they sense in an asymmetrical manner? And we thought back at the conflict of Colmer and Agder and thought, well, maybe that comes from the Reisner fiber. So Pierre-Luc Bardet, who came from Drosophila, joined the team, and he made the first CRISPR knockout for the scospondine, where he targeted the hemidomain important for aggregation, and he got a null allele and also what we thought would be an hypomorph with 5 amino acids added to the protein due to non-homologous end joining after the cut of the CRISPR. And in all those mutants, the 13 and the 15 allele, we had the same observation that there was no more fiber in the central canal. So we lose the fiber. And when we lose the fiber, what Adeline showed afterwards was that we also lost the response to the mechanical bending and the compression. So the fiber is really necessary for sensing the bending on the site. And so without going into too much detail, because now this is published, but what we showed is in fact that the fiber is under tension in the central canal. It's really like Ernst Reisner described it in vivo. We made a knock-in GFP here, and you can see the fiber is extremely straight in vivo. And not only it's straight like a ruler, but it's also interacting with the little cilia beating of all the cells in the central canal that generate the flow. And we believe that this interaction with cilia enables the fiber to be under tension so that you can have this beautiful straight line via the many weak interaction of cilia with the fiber. And so the way we understand it is that the fiber being under tension, when it's bending to one side, it actually gets closer to the cells, the CSF contacting neurons, and we get an interaction between the apical extension and the fiber, which activates the cell during contraction and not stretching, because then the fiber gets away from the apical extension. So in zebrafish larva, this is only 8 microns, and the apical extension is only 3 to 5 microns. So it means that the phenomenon is very small. In fact, the fiber movement during bending is in the range of the microns. So it's very, very small. And what we'd like to understand, and we're starting experiments in that direction, is to see whether there is a direct interaction of the cilia of the cells with the fiber, so that it looks a little bit indeed like hair cells with the Reisner membrane, or if it's more that the apical extension detect an enhancement of the flow gradient, because on the fiber, the flow, like on the wall of the canal, is 0, and therefore the gradient is larger. So we're trying to do experiments to test if there is a direct contact in this respect. So then when we looked at behavior, which was our initial interest, we looked at what happens when you get rid of these CSF contacting neurons and what happens to the fish. And what we saw, to make it short, is that not only the fish were swimming slower when they lacked the sensory system, but also we noticed by analyzing their posture, or typically zebrafish larvae are keeping an horizontal posture where the two eyes are staying in the plane. When we looked at their behavior, when the CSF contacting neurons are intact, they're staying horizontal. But when the CSF contacting neurons are either killed or silenced, we saw that we had rolling of the fish where the fish loses balance during fast swimming and they're not able to maintain their proper swimming to stay in horizontal plane. And so that's interesting because people think always about the vestibular system being necessary to keep posture. But in fact, for many animals where the body is quite large and the musculature is really important in the body, you also need to keep track of the spine position. And so we established through a series of papers, I won't go into detail, that the sensory cells, in fact, project after detecting the compression on one side, they modulate the activity of excitatory interneuron and motor neuron in order to affect the locomotion. And what is really nice is that the work of Ueno in Japan, together with also Niccolò Ziampiri in Germany, has shown that the circuit is very similar in the mouse. And that if you ablate this cell in a mouse, you also get the effect where the mouse is going to fail at doing tests where it needs to walk on a very small rod or try to jump from one ladder to the other with big steps. And so every time the animal is challenged, not only for a fish, but also for a mouse, these cells are important to maintain the fine postural skills. So now, what are the consequences of losing this sensory system? So what we found, and again, this is a summary, is that when we affect the sensory system, what the animal will look like is not straight anymore in a frontal plane. If we look at the P12 mutant that is necessary for the mechanosensing, we have like kyphosis. And now if we look at the scospondine mutant, where we have lost the scospondine and the Reisner fiber, we get even more striking phenotype of scoliosis. And so it's really interesting, and the mechanism are partially understood by the work of collaborators like Daniel Grimes and others, where URP from the urotensin 2 family and their receptor on the muscle and notochord are involved in the process downstream of the CSF contacting neurons, but they are not the only one, and we're investigating the other genetic component. So one doctor in the hospital, Pied Salpêtrière, Laura Mariardi, is working with Feng Quan in the lab to actually identify new genes from families of patients where we have recessive mutations. And what we're excited about is that we have 3 mutations that we generated knockout for in fish. And in the 3 cases, we have a very nice defect in the body straightness and also in the curvature in the adult. So we're now investigating the mechanism of that. So that's really nice because not only we can use zebrafish to investigate the circuit mechanism, but we can also use this model to actually look at postural control and how do you build a straight body, because for a fish, it's actually very tricky to do that. For a mouse, the forelimbs are helping out. The DRG are providing a lot of information to push and reduce the tension on the spine. But for a fish, it's much harder. And for a bipedal like us, it's like kind of an in-between. So we can learn from the fish to see how to really understand postural control and how posture impacts morphogenesis. Then the second short story I'd like to talk to you about is now how is the sensory system involved in detecting pathogen and can impact also morphogenesis. And so what we found is that the sensory cell that they express, like I said, a PKD2L1 receptor that has been found by other, including Charles Zucker, to be sensitive to pH and osmolarity. And so we were interested to see what could these cells sense and taste inside the CSF. And in fact, via a discussion with Hitoshi Okamoto in the RICAN, I thought, well, it could be really nice to test if they could be involved in the meningitis, because when patients have meningitis, they have this, stiffness of the neck where, we only see the neck because it's obvious, but in fact, it's the entire axial musculature that is very stiff. And so we decided with Andy Prendergast to look at this question and to ask the question of whether these cells are involved in detecting pathogen that trigger meningitis in humans. So we used 2 pathogens, and here I'm showing you Streptococcus pneumoniae. It's a bacteria that trigger as the main source of bacterial meningitis in humans. And we fused it with an M. cherry, injected it in the brainstem, in the ventricle, and then you can see that the bacteria after 24 hours is really invading the entire cerebrospinal fluid. And we used also Listeria monocytogenes, and we got similar results. So I will show you the result of Streptococcus pneumoniae. The first thing that we observed is, contrary to what I've shown you for the knockouts of the sensory system, when animals are infected, they show a dorsal curvature, which is really interesting. It's the opposite of what I've shown you when they lose the sensory system or the Reisner fiber. It's a dorsal up phenotype. And this, in fact, looks very similar, even though it's a fish, to what patients will have if you don't treat them with antibiotics, where they will do dorsal arching called opistotonos in patients. So, in our countries, we treat with antibiotics and we typically don't see that. But the next step after the stiffness of the neck is actually to start to do dorsal arching and without being able to change posture. And so we thought, well, if we get the opposite phenotype of knocking out this sensory system, it could be that it comes from the cells being activated during the infection. And so we did calcium imaging on many, many animals. And what we saw is when the bacteria is injected in the ventricle, we saw that the flickering of the cells was gone. And instead, we had very long-lasting activity of a subset of the cell that were showing very large calcium transient, typically more like 5 or 10 times what you would get in the control condition. And so was really intriguing. And so we went ahead and looked at what happens and what is the role of these cells when they are activated by the pathogen infection. And we used a tool in zebrafish that is called nitroreductase, where you can, using metronidazole, activate this enzyme to kill the cells that express it. And so we use this to kill all the CSF contacting neurons during the infection. And what we saw is that the survival of the animal was much less. And if we then used another tool to silence the release of neurotransmitter from the cells via botulinum toxin expressed just in the CSF contacting neurons, we got a similar phenotype of having less survival of the host and more bacteria in the body. So it looks like these sensory cells are in fact recruited to boost innate immunity. And we therefore look at what the receptor of these cells could be in order to detect the bacteria. And we find that many receptors were expressed. In addition to the PKD, we had TACE2 receptor, TACE1 receptor, and numerous receptors for peptides that are involved in innate immunity, where we typically find both the peptide expressed in the CSF contacting neuron together with a receptor. And so we've been testing the response of these cells in vitro, and we find that, in fact, Streptococcus pneumoniae release a lot of bitter compounds that are similar to what we taste in the tongue, where if you have bad milk and you're testing the content of the milk, you will have this bitter feeling in your tongue and you will immediately spit it out. Well, there is the same effect in these neurons where if you use the metabolite from the bacteria and you puff them on cells in vitro, you see very large calcium transient that are associated with the bacteria presence. And so they express a multitude of these receptors, in fact, to mediate this effect. And upon activation, these cells will not only influence posture, but they will also influence innate immunity and morphogenesis. And recent evidence from the lab of Wyart in mouse, and also David Julius also in mouse, is showing that these cells are also activated during spinal cord injury, and they can modulate the proliferation of cells around the stem cell niche in the central canal. So we're now investigating this question. And so to talk to you about the last part and final part of my presentation, I'd like to show you what we've started to investigate on the question of tonic immobility that I illustrated for shocks in the introduction. So a paper came out 5 years ago from the lab of Michel Arends, where they found that if you expose zebrafish larvae to a grating, and the animal is seeing a grating and trying to swim in the direction of the visual grating, after a while, if the experiment is done in open loop and the animal has no consequence and no effect of trying to swim, the animal will give up. And the way the animal will give up will be via a massive activation, of the radial glia that you can see here. So I think Misha came last year, if I'm correct, and he probably described this to some of you. So there will be like a really massive glial calcium wave that is almost like a tsunami compared to what you typically see in glia. And this will happen just before and for tens of seconds after the animal has started to enter this state of tonic immobility. And so this paper 5 years ago, almost 6 now, has been really quite instrumental for us to think, well, maybe glia is involved in controlling our behavior, and in particular in stopping the motor output in this context of tonic immobility. And so my lab has been really invested quite a lot of energy to map the motor circuit in vertebrates, and we believe that there are strong conservation. So in mouse, like in cats and in rats, mice, and so on, the motor and voluntary command are coming from a region at the boundary of the midbrain and hindbrain called the mesencephalic locomotor region. And this region projects on the reticular formation here in order to send the command to the spinal cord. And so you need voluntary movement relies really on this mesencephalic locomotor region going to the hindbrain going to the spinal cord. And when you have typically sensory inputs, they will converge onto the reticular formation to trigger an integrative reflex with 2 to 4 synapses, 4 synapses being the olfactory command. And so we started to dig into this with Maha Lakshmi Danashekar, who started to image first how the radial glia that Misha's lab had found recruited during the tonic immobility look like. And what Maha struck upon is that in this area, in the lateral oblongata, you have like very fine processes of the radial glia. And it's true as well for this region of the rostral brainstem. And this is precisely where are located the noradrenergic neurons that are not only in the locus ceruleus, but they are also, in fact, much more numerous in fish in the medulla oblongata here. And here you find all the soma of the noradrenergic neurons. They have axon going to rostral spinal cord on one side, and they have axon going towards the locus ceruleus on the rostral end here. And so there is very clear match between the noradrenergic neuron and the radial glia. And if we look at the projection here, you can see that the feet of the glial cells were really located. You see here in the region where the soma of the noradrenergic neurons are. So they have a very strong, intimate anatomical connection. And so what Maha did is first she tested many aversive stimulation, and she showed that if she's using mechanical aversive stimuli or chemical aversive stimuli like pH, she could trigger a very strong tonic immobility in the fish. And she saw that those were associated with a recruitment of the noradrenergic neurons. And so she tested how the fish respond then to only the activation of the noradrenergic neurons. And so in these experiments, to be careful, Maha enucleated the fish, so they have no eyes. They are little zebrafish larvae. She's recorded, tens together at once. We're just zooming on one. And what she finds is that if she reached a sustained activation of the noradrenergic neurons, here is for 3 seconds, she can get this complete tonic immobility that is associated with a rolling behavior of the fish, where the fish start to roll on its side, very similar to what you see in the shark video. So the animal is on its side completely now, and it occurs and lasts for tens of seconds after the stimulus. And so when she looked now at the fish from the side, she discovered something interesting, that not only the fish was modulating its motor output, but it was also changing after stimulation. Its heart rate became very low, and it triggered bowel movement and movement of the jaw just from the activation of noradrenergic neuron in the medulla oblongata. And so Maha went ahead and started to, quantify those phenomena. And she started to look at the calcium wave with a little bit more scrutiny. And she noticed the following, that when you record the calcium wave upon stimulation of the noradrenergic neuron, it seems to invade the brain and spinal cords, and it lasts for very long in the brain. So if we now look at the time scale, we see that the time scale are lasting for tens of seconds, up to 30 seconds in the brain, when it's only lasting for 5 seconds in the spinal cord. And so she's done experiments where she reproduced electrical stimulation experiment that we had done in the lab, where if we stimulate the mesencephalic locomotor region, we could trigger the forward swimming in zebrafish. And if we if we stimulate the obex region that is at the boundary of the spinal cord and the brain stem, we had the same effect. And so Maha did the same experiments, where now she compared the animal in which the opsin is expressed, and she used 3-second light pulse at the beginning here to activate the noradrenergic neuron. And she saw that in the control, it didn't impact with opsin expressed in noradrenergic neurons the motor outputs. But in the animal expressing the opsin in the noradrenergic neuron, she had this very long motor arrest where you couldn't elicit the activation of swimming like you do in the control. So it looks like the information from the brain to spinal cord is really impacted when the noradrenergic neuron triggers this calcium wave. And so Mara went ahead and recorded now from the motor neurons. And what she finds is that in the motor neurons, when noradrenergic neurons are expressing the opsin, she can see this very clear silencing of the synaptic input, where for tens of seconds, there are no more synaptic input received on the motor neuron. And it's a complete quiescence period without having any impact on the motor neuron properties themselves. So it's not affecting their biophysics property, it's affecting their input from the brain. And so in conclusion, we have evidence that this tonic immobility can be induced just by activation of the noradrenergic system. And it's a way to mimic, in fact, what happens when very strong sensory stimuli are recruiting our noradrenergic system. And such a sustained activation of noradrenaline will disrupt the postural control of the animal, will get them to reduce their heart rate, change the contraction of the bowel that we still need to investigate how it works, and then also impact the motor command so that the brain doesn't talk to the spinal cord anymore. And if you stimulate the reticulospinal neuron, you can't get any more motor output. So it's really a fuse that forbids the motor output to happen. And so with that, I reach, all the different short stories I wanted to talk to you about. And I'd like to just thank all the people who, throughout the years, have been involved in this adventure. Like Maria and Noah, we heard in the beginning, it's really fun to train people, and it's been a real pleasure to work with people from different fields. At the image of OIST, very interdisciplinary, and it's really great to be back. So I'd like to thank my hosts, in particular, Greg Stevens and Vincent Laudet and Ishiro Masai. And I really would like to thank OIST for welcoming us again. It's really a pleasure to be back.
Oh,
Get the videos of the
yes, yes.
yes. Change like the shoes. Video is only 1 minute, but you see that by caressing the nose where the electroreceptors are located, how you can actually try to really calm down and get complete rest like that. It's impressive. And trying to have the same radar clear that have positive. And my dream is to be able to actually transfer the resistance that led the shark to be completely still. It's quite remarkable. Maha doesn't know how to swim, and she's not very good. So that's for the shark tonic mobility. But you have take it back to not. See where it is. So the animal actually is about to be eaten. And there must be something that scares the predator away. The animal feels that. And after the guy is going to stay completely still. And it's quite remarkable. That's one that's pretty for even longer than to the predator is down. And then you see that the breathing is clear, and then eventually the bada is going to go with. But I think it's fascinating. And in fact, it does happen in human. People who are, on the very same spread because they can also see far so they can immediately we both really know how to relax. I think it works. It's completely fine afterwards. So I hope you got very information. And thank you very much for that.
Thank you very much, Claire. It was a very interesting talk. So yeah, if I ever find myself in a situation where The shark is presenting danger for me. Now I know how to save myself. Thank you very much. I have just like one question. So when you knock down the receptor in the sensory cells, like you mentioned that the curvature is different when you when the animal is infected. Is that correct?
Exactly. So in the cases of the PK12 mutant, or when you lose the sensory system with the fiber, you get in the case of the receptor here, you get this kyphosis where you get actually a curl down. So it's a ventral curvature. And what we get when we have the infection is a curl up curvature. The loss of the fiber is associated with an even more striking phenotype where if you see here, it's a 2D deformation, ventral curvature. Here, it's a 3D deformation. And so it's very interesting to think how mechanically you get the 3D deformation. And for that, we don't really understand yet. We see that there is a distribution of the receptor for peptide that are different on the ventral and dorsal musculature, but we don't really understand how the left-right, bending occurs. And Feng Quan in the lab has very interesting mutants where in the case of those crispant NPR3, she's actually getting left versus right defect without having the always the ventral one. So we're really curious to see how the left versus right is really integrated in the body. It's something we don't quite yet understand at the molecular level. How do you make sure once you feel like you're straight that you readjust the growth or the activation of the muscles, that the morphogenesis will not be biased toward the left or the right. Something I like to say, taking advantage of this on the morphogenesis, is that strikingly, those mutants, they have very similar phenotype with Sherman disease for PE12-1 in humans. And the bias for gender is the same. So this kyphosis occurs much more in male than in female for humans. And it's true as well for the fish. And the scoliosis occur a lot more for female than male, and it's the same for fish. So not only you have more of one gender, but you also have more, increased kind of curvature and deformation. So it looks like there is, probably a role of sexual hormones, and we find the receptor for LH in the CSF contacting neurons. So we're really curious to see how does it work, and why is it that you have defects that are different in male versus female? Not clear. Yeah.
Thank you very much.
Yes.
Anybody else has a questions?
Thank you for the amazing talk. I have a question about the last part of your talk about immobility. And so the triggering stimulus is, very different. But I wonder if there is any common mechanism in terms of implementation with sleep induction.
Oh yeah, so that's fascinating question. So in fact, Maha would really like to tackle that question for our postdoc if you're interested to hire. She's going to be on the job market at the end of the year. No, so in fact, I think it's a fascinating question because we have actually expert of sleep research in humans as neighbors now in the institute, and they have very interesting data in humans, but also in mouse from Anita Luthi, showing that you have switch of non-REM and REM sleep that occur with a change of activity in the locus ceruleus, and so there is a very interesting neuroadrenergic contribution to investigate there, and it's not clear to us how it works. So we are discussing now with people in the institute, and another Luthi, Andreas Luthi, came also to the institute recently. And what you see in amygdala during freezing is that you have, depending on the context and depending on the reward and many aspects of the experiments in terms of behavior in the mouse, you have recruitment of completely different neuronal assemblies. And so they don't really understand how it works. And so we're starting to wonder, but just collectively in the institute, whether it could be that you have actually a role of neuromodulation that doesn't occur only via noradrenaline acting on the neurons, but possibly not through a tsunami like I've been describing here, but via modulation locally of the calcium in the glial cells. And so there are multiple evidence that during freezing, you have in the amygdala large calcium increase also in glial cells. And it's really not clear whether you could have modulation of the motor output with more finesse by actually just acting, not like the massive tsunami, but just acting locally in different areas of the brain that could impact the way the neural networks function and operates in the surrounding. Yeah. Yes.
Yes, thanks a lot for the talk. When I saw like the fish that had the mutations, like the deformed spinal cord, I immediately thought about the aging fish when you do the fish raising.
Yeah.
I was wondering, like, have you looked at the ricin fiber in the aged fish?
Yeah.
Yeah.
No, so it's actually a really good point. I'm really happy that you asked this question because right now we are only thinking about this question in terms of age. But when you have big fish facility like Yuko has here, or like we have in a little bit smaller in Paris, what you notice, in fact, is that there is a very important factor, which is like whether the fish get sick. And so it's not only age, but it's actually a symptom that you can have after infection. So in fact, many facilities who have suffered from having mycobacterium or other pathogen in their facility, they notice that the sick fish will actually get deformation of the spine. And it's a correlation that in humans, surprisingly, we haven't been doing. So I have now my new director is an epidemiologist. So I'm hoping to know how to look into that more in humans. But we don't actually know if infection and previous infection could impact the way you not only have grown your spine, but also the way you maintain your spine. And so the mutant I've described for the PE12-1, it actually comes at the kind of senior age of fish age that you get the deformation. And it's very interesting to think whether it's a problem of immunity that could lead to a deformation, or if it's a direct problem of sensing. It's a very complex point, but I think it's a very interesting one to also consider for ourselves, that infection can impact your body shape. Thank you.
Where do you project to exactly?
Yes, I
and
removed that
how-
slide. Yeah, so I didn't go into too much detail because I didn't want to bore you guys too much. But in fact, it's quite fascinating how they're projecting on the rostral most ones that are important for rolling because they are the one projecting on the equivalent of the neck muscle and all the occipital pectoral motor neurons. They actually project directly onto those large motor neurons in the rostral, spinal cord and caudal hindbrain. And they also project onto like the inferior olive and key areas of the brainstem that we haven't investigated too much. What we described is that they are quite complex because like neuromodulatory neurons, they project onto the axon of many command neurons, like the Mosner cell, a big command neuron, or the reticulospinal neuron that are important for forward locomotion and turn. And we find that evidence that they actually project onto the axon. So they could release GABA onto those axons and potentially shunt the transmission of information from the soma to the synapse. We haven't demonstrated that. In the spinal cord themselves, it's very much like what was confirmed in the mouse, that they project onto excitatory promoter neuron, onto motor neuron, and they also project onto themselves. So they form a little bit of a hub of connected cells. Yeah. Okay. Well, I really want to thank you very much for coming before a long weekend. Thank you very much.